Thank you for joining us for our hot topics. Today we're very lucky to have Professor Michael Lin from Stanford. Michael received his AB summa cum laude in biochemistry from Harvard, and he followed that with a PhD in biological and biomedical sciences, also at Harvard at the medical school. He also received an MD degree from UCLA in 2004. Dr. Lin is currently a professor at Stanford, where he leads a research group developing protein-based tools for imaging and controlling biomedical processes. He has a long string of awards, and to me, most importantly, he is the author of what I consider as the most viral slide deck on COVID-19 in the recent days. Michael, welcome. Welcome to CSAIL, welcome to MIT, welcome to the College of Computing at MIT. We're eager to learn from you. Please go ahead.
Oh, great. Hello. Well, thank you very much, Daniela. Yeah, thanks everyone for taking your time to join. Yeah, it's my pleasure to share with you what I've learned myself about the virus and the disease in the last few weeks. So, we're living through unprecedented times. We've seen nothing like this since perhaps the 1918 flu epidemic. And so I think it's useful for us to all learn the facts and try to spread knowledge about the disease. So I'm going to get started and share my PowerPoint window. So some of you have seen this slide deck. Some of you have not. And I understand this slide deck I had made for my own lab meeting to basically ease anxiety among my lab members and use the facts to try to put together a plan of action. And so I'm happy to share these facts with you as well. And I'll try to explain, I'll try not to get too deep into the biochemical details, but hit the most important points along the way. So as some of you have seen, this is not a pretty PowerPoint. I haven't had the time to make it really glossy. It's not like a TED Talk. I'm not pushing a simple idea. There's just a lot of facts. But I think it's only when you understand all the facts together that things make sense. So we'll just start with some very general numbers to put some context into the numbers we're going to get into shortly. The total population of USA is close to 330,000. I've been doing a lot of my calculations based on numbers in California because it's just easier for us to think about locally. That's about 1/8 of the USA, 40 million people. So, the major causes of death for young people in the US are traffic accidents. There's 30,000 per year in the USA, about 3,000 per California. For older people, it's actually flu is the leading cause of death. There are 40,000 deaths a year. It's never really known how much, but the range this year is estimated between 22 and 55,000. So out of that 40,000, about 5,000 in the USA. So about 5,000 in California. So for a population of 40 million in California, you can use this sort of number to get a general idea of how lethal flu is. If 25% of people got the flu virus and only 5,000 died, that's a infection fatality rate or IFR of 0.05%. It's probably actually more like 0.1%. That is more like 12% of the population might get the flu virus in a given year because we have flu shots. So is everything okay? You guys can see the slides, Daniela? Okay.
Yes, everything is going great. Thank you very much. Yes.
Okay, great. So we'll get right into some quick definitions. The disease name is COVID-19. I think we have all seen this term much more than we want. Technically, it refers to the disease, not the virus. In practice, the term cases of COVID-19 are used to refer to having a positive lab test, whether or not you have symptoms. For reasons that are partly political, partly medical, if you just have the symptoms, you're not necessarily yet a COVID-19 case because you could have, for example, a bad case of flu. So you tend to wait for the lab test result. The WHO, the World Health Organization, introduced the name COVID-19 after holding the world in suspense for a few weeks, saying how important it was to get the name right. And then when they announced the name, there was no name for that was the name for the disease, but there was no name for the virus. And so everyone started using COVID-19 as the name for the virus because there was no other name given by WHO for it. So this has led to some confusion. A lot of people use the term COVID-19 to refer to the virus. I mean, I guess that's fine in a popular lay point of view. But technically, it actually makes a difference because I'd like to talk about cases of the disease and infection numbers based on an estimate of the virus. And so I try to use the virus, a different name for the virus when possible. I mean, I personally think COVID-19 is a terrible name because it stands for Coronavirus Disease 2019. And WHO says if you want to talk about a virus, you could call it the COVID-19 virus. But that would mean Coronavirus Disease 2019 virus, which is a mouthful for saying nothing. It basically reveals that the disease name has no informational value. So previously we had named diseases by some sort of description of the disease. So SARS was named SARS because it was called, because it stood for severe acute respiratory syndrome. And actually this disease is very similar to SARS, both in symptomology and in the agent, in the agent that causes it. So the World Health Organization could have just named this SARS-2, and that would have been both accurate and descriptive. So what is the name of the virus? It was initially called 2019 nCoV, that stands for 2019 new coronavirus, and it was given by some infectious disease organization. This hasn't been picked up because it's a really hard term to remember. You don't usually think of numbers when you think of naming things. So, and I think it's also misleading because it gives the impression that this virus is actually novel. It's not particularly novel, and I would say it's the least novel of the new viruses we've isolated and sequenced. The most interesting thing about it is that it's not that novel. And that's because it's 96% identical to the virus that causes SARS in 2003. So in fact, the biological name for it, the name for its sequence is SARS coronavirus 2 or SARS-CoV-2. So we'll use this name throughout my presentation. And I like this name because it reminds us that SARS-CoV-2, reminds us that SARS-CoV-2 is very similar to SARS-CoV-1. And so a lot of the things that we've learned about the coronavirus 1, which came out in 2003, so almost 20 years ago, is very relevant to predicting what would happen with SARS-CoV-2 and COVID-19. So I would just say, if you haven't realized it yet, I'm not a big fan of how WHO has managed this crisis, but WHO decided not to name the virus SARS-CoV-2 for precisely this reason, because it made people think of SARS. They wanted to obscure the relationship between the two viruses. They thought it would be unfair to call the virus SARS-CoV-2 and to call the disease SARS-2 because it would remind people about SARS. Actually, I think that would have been a good thing to do. And as scientists, it's better to be clear about what you're working with rather than trying to create false distinctions. So I call it SARS-CoV-2. So what are coronaviruses? What are SARS-CoV-2? I don't have a picture. I mean, I think you've seen many EM images, many illustrations of the virus. Like other viruses, it looks like a little round ball with a little spike sticking out of it. From the molecular biology point of view, what's interesting, important to know is it's fully RNA. It doesn't go through a DNA phase. And that we're actually very familiar with this class of viruses because they account for 10 to 30% of the common cold. And they're unusually stable for RNA viruses. So we're used to hearing that RNA viruses mutate very quickly. RNA viruses will always be less stable than DNA viruses, but among RNA viruses, these seem to be pretty stable. For example, the coronavirus OC43 causes the common cold. It was sequenced once in the 1960s. It was sequenced from isolate from the 1960s and is sequenced again from isolate in 2001. And those only had 2 amino acid differences, 6 nucleotide differences. So that's pretty amazing. As you've probably heard, there are many coronaviruses in bats, and they seem to hop easily between species. So they're quite adaptable to a variety of mammalian species. So we had the Middle East respiratory syndrome, MERS virus, that hopped from camels to humans. The original SARS virus hopped from bats to probably the civets and then humans. And then this one most likely came from bats as well through some intermediate animal. And there's a bat virus that's very closely related to the SARS virus. And you can see that in the bottom red arrow. And I made this point that the human cold virus, OC43, is very closely related to this mouse hepatitis virus. Actually, what's not shown here is there's also a cow virus that's very closely related to humans. So either humans or cows seem to have given laboratory mice hepatitis. So, so how might you kill this virus? Everyone wants to know how do you kill it? So it's an envelope virus. It has a thin plasma membrane that's only 2 molecules of lipid wide. So that's actually really easy to kill. Anything that would dissolve a lipid will kill the virus. That includes soaps or detergents, solvents, organic solvents like ethanol, isopropanol. I like to use Windex, which is detergent, and bleach will kill it because it just kills all proteins and biological molecules. So it's also sensitive to temperature. I mean, I should say that the original SARS was sensitive to temperature. So the current SARS too is likely as well. And there's some numbers that you can look at. For example, on the bottom left, this is a graph of how much titer reduction you get with different temperatures. Oh, I left my temperature numbers off. That's actually, I think 37, 33, and 28. I'll have to put that back on. So, and then the survival of the virus depends on the surface. So this is a paper that was recently published in New England Journal of Medicine. On smooth surfaces like steel and plastic, it survives for longer than on porous materials like cardboard. So that means on paper, towels, or napkins, the survival should be like cardboard or lower. But also the viruses will get trapped in these paper fibers, so it's not easy to get virus out of a napkin, for example. And then it can be killed by UV. So the amount of UV and sunlight is enough to kill, probably is enough to kill SARS-CoV-2 by 90% after about 3 hours.
Michael, could I ask you a question?
Yes.
So I heard that the virus can survive up to 9 days. The data here does not take us to 9 days. Can you?
Yeah, the data here takes us to 3 days. So most headlines will say virus can survive up to 3 days. So you could see, yeah, thank you for the question. You could see here on the stainless steel and plastic that there's, the dashed line is the detectable amount of virus. So you can see that at 72, you still are a little bit above detection. And then 96, it's undetectable. So, so a lot of places, a lot of news headlines will say 3 days. That's true. But that's a 3-log reduction, so that's 1,000-fold. I think it's fair to use, it depends what you want to use, 10-fold or 100-fold. I use 10-fold because I think of transmission risks as significantly lower once you've had a 10-fold drop.
So I have a follow-up question, if you don't mind.
Yes,
so we know that there is a shortage of masks in hospitals. So if we look at this data, does that mean that if we save the masks for 4 days, then they can be reused?
Yes, correct. That's right. They'll accumulate dust and other things, but as long as you can breathe in them easily, in theory, you can just set aside the mask and wait 4 days and pick it up again. Yes. Okay, so should I move on?
Yes, please.
All right, so yeah, so now that we know how to kill the virus, we might ask how does SARS kill us? So as many of you know, you get initial flu-like symptoms, sore throat, cough, fever, and aches, shortness of breath. And for many, in fact, probably most people, they only have these symptoms. The symptom onset is 48 days, so it starts about a week after infection. In severe cases, it will spread to lungs and cause pneumonia, and that's shown in the top image. So this can be diagnosed by CT. There's a wide variety in symptom severity. So 50%, about 50% are estimated to be asymptomatic. That's based on data from the Diamond Princess, the ship in Japan. 5% will require hospitalization and supplemental oxygen due to difficulty breathing. And that 5% is taken from sort of estimates based on diagnosed cases of 10% elsewhere, adjusted for the 50% rate of asymptomatic presentations.
Michael, we have some questions, if I may interject. So one question is the disease severity related to the dose of virus exposure?
Yeah, that's a really good question. I suspect it is. I haven't seen any data because obviously you can't dose people with different amounts of viruses, but I suspect it is. Perhaps what might happen is if you get a lot of virus at once, you'll have more immediate infection in the lungs, and you'll just start off with a higher viral titer, and then your immune system is behind in the race. And so, example of this is the doctor who raised the alarm in Wuhan. His name is Li Wenliang, I think. And yeah, it's funny. It's such a sad case. Every time I say his name, I start crying. But because he was only 39 and he died, so that was really unusual. He was treating multiple patients with the virus, so it probably was because he was breathing a lot of virus.
Okay, a few more questions, Michael. So, does one maintain immunity to the virus if they go through it once?
Probably. I mean, there's no data that suggests that you lose immunity. So almost, if it's like any other virus, then it's likely that you are immune. I mean, SARS patients have proven to be immune. So I think we should work with that assumption here.
Also, when you get, how long after the infection will you test positive? In other words, you're infected today, you show symptoms, let's say 8 days from today. At what point will you test positive?
Yes, you will probably test positive around 4 days, 5 days. Yeah, so these are great questions. So what happens in the course of the disease is the virus will land in your throat and it'll start replicating. And it's now a race between your immune system and the virus. So the virus will, it might double, or it might, well, it's not doubling. It's probably, it probably has a burst size of several thousand, meaning one cell can put out several thousand viruses. It will do that over maybe 18 to 24 hours, so about 1 day. So the virus is replicating really quickly. For you to get a positive PCR test, you need to have a swab that gets enough virus, then creates enough nucleic acid in the reaction and for the test to detect it. So my guess is that you will need to have the virus for 3 to 4 days before you have a positive test.
I will have one more question and then we'll pause. There are so many questions on the chat. I will ask you one more question. We'll come back to other questions later so you can go on with your presentation. And so the question is, what is known about the disease severity? Hey, Jill, I was just reading a question and I lost it. 1 second. Sorry.
Oh, I have a question. What is known about its disease severity with respect to AMHC alleles?
What is known about why some people are more or less susceptible to catching the disease?
Yeah, right. That's a good question. So some of it might have to do with MHC alleles as described. Although, right. So yeah, it could be that. It could be differences in innate immunity. So your body's first reaction to virus is to produce cytokines and and then have cells that are infected with the virus actually shut down protein synthesis so that viruses can't reproduce as quickly. So it's kind of like a fire alarm, the first step. That might differ between people. You know, you wonder why kids don't have severe effects from the virus. So perhaps kids have a stronger innate immunity response. And it's only a few days later that your T cells arrive your T and B cells um, are activated, and the antibodies are produced by your B cells in sufficient number to block the spread of the virus. Um, so the fact that some people come through it without any symptoms at all, like kids and like 50% of the population in general, might have to do, my guess is has to do with differences in innate immunity. And then how effective you are in having your adaptive immunity, so that's your T cells and your B cells, that specifically recognize the proteins on the virus. How successful you are in getting those working and clearing the virus may have to do with MHC alleles, may have to do with how many white blood cells you have. Certainly, it seems to be a more of a problem as people get older.
Thank you. We'll pause. We'll come back to questions after we hear more of the talk. Go ahead.
Sure. Yeah. So when it gets really bad is when you need mechanical ventilation, and that's about half the cases that require hospitalization to begin with. And then, you know, when people die of the disease, it's due either to direct breakdown of the alveoli in the lungs, and there's a photo of that in the bottom right, or you have this massive release of cytokines by your immune cells because now you've recruited billions of immune cells to the lungs, and they're all trying to fight the virus, and they release a lot of cytokines, which actually can cause low blood pressure and multi-organ failure, especially kidney failure. So throughout this, you know, this was a slide deck that I put on the web, and I have links for all the facts, you know, just to make sure the facts are double checked, and for more information. So if you go to my Twitter feed, which is Michael Z. Lin, all one word, Z as in zebra, Michael Z. Lin, you can download, it's a pinned tweet, you can download the slide deck from there in the future if you want to browse through any of these links. So yeah, it doesn't make for a pretty presentation, but I think it makes for a more informative document. All right, so now a lot of text. So what everyone wants to know is, what is the probability I will get the disease? Like, how many people have the disease? And that's impossible to know from looking at the case numbers. So this is something where, you know, the news will report the case numbers. It's misleading. The case numbers are always less than the infection numbers. So if you want to know infection numbers, you have to, I think we have to go by, we have to estimate the probability of death and work back from there. So how do we know the probability of death from infections? Well, you could start with some good data. So you could start with a place that has done the most testing per capita, and that's South Korea. So right now in South Korea, what you can see in this graph in the middle is the number of cases diagnosed in orange, and the number of deaths is in red. So it looks like there's a small number of deaths, but actually we know that deaths on average lag diagnosis by 2 weeks, and this means infections by 3 weeks. It's actually 2 to 3 weeks, or infections by 3 to 4 weeks, but diagnosis tends to happen around 1 week after infection, when the symptoms first start. So if you look at the death and case curves, the death curves indeed lag the case curves by about 2 weeks. So this case curve, as you can see, is sort of flattening out. It's reaching sort of a constant rate of about 100 per day. But the death curve looks like this curve from 3 weeks ago or 2 weeks ago, which is right around here. So it's still sort of in this inflection point. It hasn't yet flattened out. That's the death curve I just mentioned. So the current total deaths occur from cases diagnosed 2 weeks or earlier ago. So if we have 75 total deaths last week, and out of 4,000 cases 2 weeks earlier, that's a case fatality rate of about 1.9%. But we know that infections have to be at least greater than the number of diagnosed cases. So that means the true infection fatality rate is less than 1.9%. And this depends on what fraction of infections were diagnosed at, 2 weeks ago. So it's somewhere below 1.9%.
Could I ask you something about that, Michael?
Mm-hmm.
So I've been watching the Johns Hopkins real-time cases website, and they report that for the resolved cases, the rate is more like 10%. How should we think about that? The closed cases are the cases that ended either in the patient being fully recovered or in death.
Yeah. Well, I think they're just slow to resolve the cases. It takes about a month if you have a severe case, it takes about a month to fully recover, free of symptoms. And these resolution numbers, I'm not sure how they're done. They might be waiting for people to be free of symptoms, or maybe they just don't follow up in a large proportion of cases. My guess is they're not following up. It's just people are too busy dealing with the new cases to worry about what happened to the old ones. So yeah, the case closure numbers, resolution numbers have been low for all the countries. So how else can we get this IFR, this all-important infection fatality rate? So an analysis of Chinese data gave an IFR of 0.5%. Some people doubt the reliability of that data. I haven't looked at it myself, but analysis of the Diamond Princess, that's a ship in Japan again, gave 1.2% as the infection fatality rate. Now on the Diamond Princess, some patients may have been helped with this new medication remdesivir, but also those passengers were on the old side, and we know old people are more impacted. So I think the 1.2 is sort of a good estimate. Also, you could look at population distribution. So if you look at the number of people over 60, there's more in Korea than in the US, and there's more in the US than in China. So, so I would guess that the IFR for the American population is probably around 1%. So an estimate based on age-adjusted data from China also gave a very similar number of 0.9% in a range of 0.4 to 1.4. So that shows you, the range shows you how difficult it is to really peg a number, but let's just go with 1%. All right, so we'll move to the next slide. Oh, I need to click, okay. So as everyone knows, death is much more likely for an older population. So that's unfortunately the case. It's not that, the same was true with flu. So it's not a surprising fact. But you can see that unlike the flu, it seems to be much worse. Now this again, I think is actually death rates out of total diagnosed cases without a 2 or 3 week time lag. So this might be actually a little bit of an underestimation, meaning that the denominator cases might be smaller, might in actuality be smaller than shown here. So this makes the disease much more severe than flu.
Michael, we have an interesting question.
Right.
Is there any reason beyond age distribution why IFRs would be different across countries? So we've heard that in the US, mostly younger people are experiencing severe disease.
Yeah, some young people experience severe disease, but I think it's still a small proportion out of the total infected in the young people. I haven't heard of good data for differences in the age distribution of severity. There certainly will be some country-to-country data. In countries where older people are more likely to smoke, they ought to be more impacted. So maybe that's a little bit of a difference. Like maybe in South Korea, older people had a smoking history, whereas that's less likely in the US. But there, I think we lack good data on that.
Also, can the virus mutate into being more deadly? We've heard that maybe there are two strains and there are worrisome numbers in France. It's not clear if there are more strains. What
Right.
can you share about that?
Yes, it can mutate to be more deadly in theory. There's no particular reason to think that it will get selected for that though. Meaning viruses actually don't want to kill their hosts too quickly. They'd rather the host stay healthy and just be coughing a lot. There's no reason to believe, there's no reason in particular to be very afraid of that yet, right? So meaning the virus is known to be stable. It's known to be, it's known to adapt to a bat host in a stable manner. I don't think there's any reason that it would be selected for increased fatality. Right, so I made an estimate of how many new cases. This is, we don't have to go through the numbers, but basically you take the current, I split it up into weeks. I go week by week. I take the number of deaths in the last week and I multiply by 100 to get a number of infections that happened 4 weeks ago, in the week ending 4 weeks ago. So, and I do this at the end of each week to update the slide deck. So this smooths out day-to-day differences in the reporting of deaths. And it also doesn't require any calculus. It's basically a numerical analysis. It's like doing Riemann sums with a time interval of a week. So what you need to know is sort of the rate of new infections each week. So if there were, for example, in California, there were 10 this week, 10 deaths this week. That means there were 1,000 new cases 4 weeks ago. With a doubling time of the infection of 7 days, some people have estimated 6 days. I just use 7 so we could sort of make the analysis simpler. With a doubling time of 7 days, then from 4 weeks ago, we're now 16-fold higher in the rate of new infections. So there are probably 16,000 new infections the last week in California, which is 1 in 2,500 people. So, and then in the Bay Area, we're more impacted in other regions of California. So I just assume it's 1 in 1,000 in the Bay Area. Now, what can you do? Or you're in Boston, which is also heavily impacted. So it might be also something like 1 in 1,000 people just got the virus in the last week. So what does that mean? Well, 50% of people will be asymptomatic, so they'll never know. And this is based on, again, the Diamond Princess data. It's also based on some mathematical analyses of the China data and of data from passengers on evacuation flights back to US. So 50% of people will never know. The average incubation is 7 days, but the transmission may begin 2 days before symptoms. So that means a large portion of people will start transmitting the virus without knowing, even if they do eventually get symptoms. So, so that means you have a risk of 1 in 1,000 from direct personal contact, perhaps, and higher than that from touching objects that are commonly touched, which are called fomites. So those are things like door handles. So in the risk of touching fomites or contracting it from touching fomites is in proportion to the number of people touching that between cleanings. So if 100 people touch that door handle before you touch it, now that risk is increased to 1 in 10. So that means we had to take action to reduce the risk of acquiring the virus, either directly or from touching. So what would happen if we did nothing? So obviously exponential growth will continue with a doubling time of 1 week. And you can do a quick, simple model as shown here. This is a very amateur model. Most of you can do better models. Again, I didn't want to do calculus, so there are no equations. It's just doubling. And then we reach a point where we run out of people to infect. And according to this model, that's in late May. And California officials estimate 56%, total infections. Angela Merkel famously said 70%. So something, between 50 and 70%. At that point, the virus runs out of targets, and it's harder for the virus to find new people to infect. Everyone else has been immune. So that makes a top infection rate of about 20% for the last 2 weeks. And then 4 weeks after that, we'll have deaths that result from that 20% infection rate. And that's huge. So that would be, if you accumulated all the deaths, those numbers shown here are California, you have to multiply by 8 to get US. That would be 2 million cumulative deaths. Again, this is really just an amateur back of the envelope calculation, but I think it's better than other back of the envelope calculations that you've seen because it uses reported death numbers and this estimated IFR. Professional models say 100 million cumulative infections. That's 30%, or 50, or you can say 56% in the case of the one that was done for California. An estimated IFR of 0.5 as a conservative estimate. So I think my estimate is not too far off. It might be the worst case scenario would be the 60% and the 2 million, but it could be better. And there's some nice sites for following case numbers and fatality numbers if you want. So compared to the flu, if we're looking at which had a cumulative infection rate of 27% and an IFR of 2%, this is probably similar in total numbers, but the flu was famously devastating. The disease itself is probably worse now than the flu because back in 1918 they didn't have ventilators or drugs or antibiotics. So this is why we need to famously flatten the curve. There are estimated an estimated 5% will require hospitalization, and 2.5% of those will require ICU and ventilation. And the average hospital stay is 3 weeks. So that means that in the last 3 weeks of May, we'll get 10, 20, and 20% of the population infected. And then that 4 weeks later in June will result in 1.6 million, 3.2 million, and 3.2 million people needing hospitalization. And due to the 3 weeks that they need to stay in the hospital, that's 8 million overlapping at the same time, compared to 1 million hospital beds. So that means we should slow down the doubling time from 1 week to at least 8 weeks. Of course, the slower, the better. So that makes the next month critical, and that's why California is shutting down, and Boston, I think, has shut down. So because we need to know how far down we can get the R0, and which is the reproduction number, or increase how much we can increase the doubling time. And due to the delay between infection and diagnosis and hospitalization, it will take us several weeks to know if it's really working. And the hard deadline for this is really early May. I should update the slide. I think it's no longer May 16th. I think, let me go back. I guess you could say it's around May 15th. It would be bad, but it won't require martial law yet. But if you got to May 15th, you would have to basically confine people to their homes by threat of arrest, because otherwise any further infection would overflow our medical system. So what can flatten the curve? So I'm optimistic that weather will help. It's not going to help a lot, but it's going to help a little bit. So based on that graph from earlier, a 10-degree rise per Fahrenheit in Fahrenheit will increase the doubling time, will lower the amount of virus survival by 2-fold. So I think I would assume it would increase the doubling time 2-fold. So actually, I correct myself. A 10-degree change, I think, increases the amount of death 10-fold on the virus. So I think it should increase the doubling time at least 2-fold. And then it should also get people outdoors, so keeping them apart. So that's why the flu is seasonal, for example. So the goal is to reduce the reproductive number R naught, which is an amount of people infected by each patient, to 1.0 or lower. The current rate is estimated to be 2, and that infection, the sequential infection time interval is about 7 days. Basically starting from around when shedding occurs at 5 days to when symptoms start at 7 days. So if one person infects 2 others in 7 days, that would explain a doubling time of 1 week. A drop in R naught to 0 will make the doubling time infinite, and drops in between 1 and 2 will increase the doubling time in proportion to the drop in R naught. So, we have been able in other countries to drop the R naught to 1.0 to a constant rate of new cases, a constant very small rate of new cases in South Korea, Taiwan, Singapore, and Hong Kong. And that's actually without shutting down the economy. That's by doing social distancing, very, very robust testing and tracing, and very strict hygiene, especially use of face masks and being very careful what you touch.
Michael, I wanted to follow up with a couple of questions from the chat. People are wondering, Wuhan was very successful in flattening their curve. Why are the rest of us struggling?
Yeah.
What should we be doing more?
Right. I think it's too early to say. In California, we just implemented a lockdown on Tuesday. It will take, and the cases are ramping up. So a lot of the increase you see in the case numbers is just due to testing. And that's probably true even in places like Italy, where they've had a lockdown for a little bit longer. So as a reminder, if I was to get infected today, I wouldn't show symptoms. If I was to be infected today, I wouldn't, and if I were to show symptoms, I wouldn't show symptoms for another 5 to 7 days. And currently there's a delay in the US testing, so I probably wouldn't be tested for another 7 days after that. So now there's this 2-week delay between when infections start and when testing is complete. So we won't see any change to the R naught or the doubling time, at least for another 2 weeks. And in fact, most people from news reports, it seems like most people with symptoms are being told to stay at home until it gets and to see whether they get better or worse. They're basically not testing unless they get worse. And so that means a large proportion of mild cases are being missed right now. As testing expands, it expands to those mild cases, or expands to people who are just household contacts, and then that's going to explode the number of diagnosed cases. So that means we're not going to see a reduction by looking at the case numbers. The only way we could see a reduction, I think, is looking at the death numbers, because those will follow the infections by 3 to 4 weeks, but that's still a faster time window than the ramp up of the testing appears will be.
Michael, lots of other questions and activity on the chat. Let's see. Can you make a comment about face masks?
Yes, we're getting there.
In Asia says you must use face masks. Masks is the only way. In the US, we're told face masks don't help at all.
Yes, I have 4 slides on face masks.
Okay, we'll wait on that then. Okay, let's see. One more question. What is the best case scenario? You told us the worst case scenario.
Right.
What is the best case scenario for recovery?
Yes, in terms of numbers, I assume, right. I mean, the best case scenario for a person.
And numbers, yeah.
Yeah, right. So the best case scenario for a person is asymptomatic disease. The best scenario for recovery without vaccines is probably something like the next line in the slide I was going to get to, which is complete household isolation. And
but for the country, Michael, for the country,
for the country,
what's the best? Let's say everyone stays at home. What's the best case scenario? How many more weeks to recover from where we are?
Right. So I think if everyone stays at home, it's likely we can limit the R naught to something like 1.0 on average. Maybe even lower. So in Wuhan, they did complete household isolation, which is similar to what we're doing now. in California, except they probably have fewer exceptions for vital industries. So here in California, we have exceptions for a whole slew of essential industries, including marijuana dispensaries. So but in in Wuhan, they had complete household isolation, and then they took infected household members to central quarantining locations or hospitals, and they practiced strict hygiene. And this dropped the R naught from 3.9 to 0.32. And now Wuhan or China is reporting no new cases. Some people don't doubt that's actually true, but regardless, they have a large drop in the number of cases. So if we practice complete household isolation, then the disease would go away. But that would require complete stoppage of even essential industries. So I don't think that's likely in the U.S., but it's possible we can drop the R naught to something like 1. And then once better drugs or vaccine comes along, then it would be safe. A big fear is pre-symptomatic or asymptomatic transmission. In Wuhan, when they took the symptomatic patients out of the home, the R naught fell from 1.3 to 0.32. So this suggests that only 24%, or that fraction, 0.32 over 1.3, of transmission events occurred before symptoms. But another study estimates that 44% of transmission is in the pre-symptomatic period. That's done by modeling. So, I'm not able to evaluate those models or the quality of the data behind those models. So I don't know how good those numbers are. But we can, we can assume something like 24% to 44%, or maybe a third of transmission events occur before getting symptoms or without symptoms. So how about completely asymptomatic patients? So the undiagnosed carriers are estimated to be about 50% as infectious as diagnosed cases. The undiagnosed carriers in this study, which is just published in Science this week, include people who will eventually get the disease. So that's not completely asymptomatic, but undiagnosed carriers are more likely to be asymptomatic than diagnosed ones, obviously. And 50% of people who get the disease seem to not have any symptoms at all. So this implies that asymptomatics are not as infectious as symptomatic people. So we'll go to the next slide. Yeah, so how effective is this? We talked a bit already about the China, South Korea, Taiwan, Singapore experiences, but you could see that here on this graph. The y-axis is log graph, is log scale. So that means a line would be exponential growth. And so you could see that some countries are still on a line, like France, like Japan actually, and the US, but others have become stable. Again, this is case numbers, so some of the initial growth in cases, especially in the US, is just from faster testing. So everyone wants to know if there are drugs. So this is the part where I probably will go quickly because this is mostly done for a biochemical audience, but I'll say that there are a few drugs that have that are very likely to work. So one is this drug that's already approved in Japan for pancreatitis. It's an inhibitor of a class of enzymes, proteases, that are required for entry of the virus. And what's interesting is it was tested in cell culture, and it has wide ability to block the replication of all sorts of coronaviruses, including SARS-CoV-1 and SARS-CoV-2. And that's shown here in the bottom left. It's also been tested against the original SARS virus in mice. So it's always nice to see things tested in mice, because the main barrier to a drug being effective is getting high enough concentrations in vivo and not having any toxicity in vivo. Things that are toxic to humans often show up in toxicity in mice, so it's a good proxy for humans. And so you could see, you could see here that this drug, chemostat, reduced the amount of death from SARS-CoV-1 infection in mice compared to nothing at all. So another one is this anti-flu drug called Avigan or favipiravir. And this is a drug that gets converted into a ribonucleoside, which is the building block of RNA. And so then it gets used by the RNA, the virus RNA polymerase, and the virus is now unable to replicate its genome, or at least not able to replicate it correctly. The idea is it creates so many mutations during replication that the virus eventually fails to replicate. So this, again, works against the current virus in human cells. And then it actually seems to have really poor potency. We use concentration required for 50% inhibition as a benchmark for potency. This number is actually really high. You usually want it to be below 1 micromolar. This is 62 micromolar. But actually, you can load this drug to a high dose in humans, 400 micromolar in your blood, which is really high, with just a pill. So this is really efficient. And it was declared 2 days ago to be effective against the virus in controlled trials by a Chinese authority. No data was provided, so we don't really know how good the data was, but it sounds promising. This one, Colectra down here, is a HIV protease inhibitor. It was thought that maybe because the SARS virus also uses a protease. A protease is an enzyme that cleaves other proteins, and it's required to process one big protein in the virus to many different parts, individual parts. It was thought that, it was thought that maybe this HIV protease inhibitor would be useful. And there was anecdotal experience that it was useful. But it turned out, just announced 2 days ago, and I haven't had time to update the slide, and I will right after the presentation, that the controlled trial failed. And so that shows the importance of doing controlled trials and not relying on anecdotal evidence. And I always thought that this rationale was very weak, so I'm not surprised that it failed. Chloroquine is getting a lot of press today, and I think it's long overdue press. It again is shown to inhibit viral replication of the current virus in cell culture. And a whole month ago, the Chinese Health Ministry said that it was superior to control treatment in multiple trials in China. But again, no data was shown. And actually, I'm not sure why they don't show data when they make these announcements. It's a little strange to me. I actually, yeah, there's cynical reasons that they might not be doing it, but for some, or it could just be a cultural thing. But in any case, they haven't shown the data, but they said they made the announcement that that it was useful. And then just 2 days ago, basically the same conclusion was reached by a French group. They showed that it was effective in reducing viral titers in patients in an open label trial. But chloroquine and its relative hydroxychloroquine, they're both similar. They're both anti-malarial drugs. They do have some side effects, so which can be serious, but they have been safely used for preventing malaria. You know, I think it's funny. You know, it's funny when the press says, "Oh, it's not proven." Yeah, maybe we shouldn't use it yet. I'm like, well, you don't have to wait for FDA approval. It's already a drug that you give for malaria prevention. The current disease is a lot worse than malaria in some respects, so I don't see why we wouldn't just go out and use it. So a drug that gets a lot of press is remdesivir, and that's because it's probably the most specific of the current antivirals for SARS-CoV-2. So remdesivir, it's another prodrug that gets converted into a ribonucleoside and is used by the RNA polymerase. It was originally designed to inhibit the RNA polymerase for another RNA virus, Ebola, but it was shown in a published paper to inhibit the SARS-CoV-1 RNA polymerase and the virus replication in mice very effectively. It also works in human cells on the current virus. And that's not surprising because the two viruses are 96% identical at the amino acid level. So for most purposes, you can think of SARS-CoV-1 and 2 biochemically as essentially the same. And so now it's being tested in humans in multiple trials in the US and in China. The China trial data are due in a few weeks. And it's gotten a lot of good anecdotal results. But again, we should not rely on that. That's why we have the controlled trials. So there's a lot of text, but basically I should just note that there are drugs just to treat the symptoms and not the virus. So death is often from cytokine release syndrome, as we mentioned, and there are drugs for that. And so China has approved one of them, which is a Roche antibody against a cytokine to prevent cytokine release syndrome. There's poor evidence, but some evidence like COX-2 inhibitors, that's just Vioxx and Celebrex, which were previously prescribed for arthritis, can prevent cytokine release syndrome, but no one's really brought that up recently. Again, like treating this is good, but it's not as good as preventing the virus in the first place. There's this big raging debate going on in the medical community about ACE inhibitors, and I'm not going to cover it because I think it's really besides the point. This is, you know, internal medicine doctors love playing with hypertension drugs, and ACE inhibitors are hypertension drugs. You know, some of you are probably on 3 hypertension drugs. I mean, this is just, internal medicine doctors live for doing this stuff. And they're fighting with each other over whether ACE2 having more or less ACE2 activity is good for the virus. I think it's a sideshow. They're talking about preventing lung injury or decreasing the number of receptors for the virus. I think it doesn't matter. I'm not even going to touch on it. So I'm not even going to cover it. But anyway, there's a link here for more clinical trials if you're interested. So there's a whole bunch of new things. You can make antibodies from patients, or you could screen antibodies in libraries in vitro and just produce antibodies that will bind to the virus. So this has already been announced by 2 companies, Vir and Regeneron. I'm sure there are many more companies doing this. This is a tried and true method. This is why when sometimes you get a travel shot of immunoglobulin before you go traveling for different viruses. So it can be quite effective. And so you can also make more drugs based on the ones that we already know, like that flu drug, like the remdesivir. And you can screen for drugs either in vitro or virtually on a computer. So all these things are being done. And then, of course, what we really want is a vaccine that we can give to the entire population. And the fastest right now are these RNA-based vaccines where they just inject RNA into your blood. And some of it goes inside your cells, and your cells will produce viral proteins, and you'll elicit an immune response to those viral proteins. So there's some more, there's a nice, for chemists, there's this review of all these different approaches. I wonder why we don't just fix the virus with formaldehyde and inject it, which is how the first polio vaccine was made by Jonas Salk. And a fixed SARS-1 vaccine was found to be protective in monkeys. And this has been proposed for SARS-CoV-2. It's not like nobody's thinking about this. So I'm guessing it's been done in China. It's been proposed in this article that was written by a Chinese scientist. So I'm sure it's being done in China. But you don't hear anything about this in the US. You know, the CDC has its own lab, and they used to do their own research. I don't know if that's been cut recently. But if they're still doing that, this is what they should be doing, just making the fastest, simplest vaccine and telling us that they're doing it. So that gets me to the next point, which is, you know, I should probably skip the many failures of the CDC and the FDA. It's interesting, you can read about it. I need to look at the time. Oh, we're out of time, so let's skip this. You guys are interested.
We are, Michael, we are still interested, and we're still here. Those of us who can stay online will.
Yeah.
And so if you still have a few minutes, we'd love to hear.
Yeah, we're gonna get to face masks, which is what was asked. So here's a little table of the symptoms of COVID-19 versus the other things. So it's useful for knowing how it's different from the cold or allergies. But basically, you don't tend to have the runny nose or sneezing, but you have all the flu-like symptoms of a fever, cough, shortness of breath, headaches, aches. So recommendations for hygiene. There are many recommendations, but essentially there are 2 types. So one is, the first type is being careful to keep the things you own in your own hands clean. So I'm a liberal user of hand sanitizer. So the alcohol in hand sanitizer will kill the bacteria. So you hear these things like wash your hands often, don't touch your face. Like, what does it mean? How often is often? Why can't I touch my face? I'm going to touch my face right now. It's totally fine. Somebody freaked out the other day when I did that. I'm like, I'm at home, everything's clean. So basically, you should establish clean zones. This is similar to like if you ever done surgery on animal or watch surgery. Obviously, you sanitize before surgery, and then you have a clean zone. Once everything is clean, clean things can touch other clean things. So that means that you should sanitize your hands right after touching things that are touched by others. Um, you should preferably only use clean hands to touch other people's things, or handling things to others, but clean your hands again afterwards. And that means also sanitizing objects you get, right? So like someone drops off something for you, I would actually sanitize the outside of the package. If it's cardboard, I wouldn't worry about it too much. I'll just leave the cardboard there. But if they're giving you a smooth object like, you know, yeah, like a CD, you know, something that can hold the virus, I would wash it. And sanitation means washing with soap or using hand sanitizer or using Windex or using bleach. And so again, if you already have something clean in your house, you don't have to worry. You don't have to keep on rewashing it, or you don't have to keep on rewashing your hands when you're at home, right? So people ask about disinfectants, you know, like Lysol or things that have bleach. These are made for large areas that you can't really soap and wash or put alcohol on. If you can use soap or alcohol, you don't need to also bleach something. Like no one tells you to put your hands in bleach after you wash them with soap and water. So if you can wash an object with soap and water, you don't need to quote unquote disinfect it with a disinfectant. So, right, so I just keep my hands to myself as much as possible. Face masks. Short answer is yes. So you'll see, so in the US, we're fond of saying that they're ineffective or that you shouldn't buy them because caregivers need them. But both of those statements cannot be true simultaneously. If they were ineffective, then the caregivers would not need them. So they are effective. So they block 95% of viruses if worn properly. And that's because viruses are carried in droplets. And the masks are filters. So, you know, there's a study done between surgical masks, which are the simple 3-ply masks that look like they're made out of paper and cloth. And then there's the cup-like masks, the N95 respirators. The study said they're actually similarly effective, but the N95 respirators I thought to be more effective. So my recommendations for mask use are based on relative risks. The reason we've been told not to use them is to save them for the caregivers, and because the risk that you'll run into somebody who's emitting virus in public is very low. So, you know, basically 1 in 1,000 at the moment. So if you're just walking by yourself in public, you'd be wasting a mask. But I think they would be useful when you're in proximity with strangers. So when you're riding in an airplane, a train, in an Uber or Lyft, because you don't know if the driver may be contagious, and especially if you're going to go to a hospital or a clinic. So
what about the store?
Yeah, so it depends how crowded the store is. I avoid stores, so I don't know how crowded they are. But I think if you're going to, I would have a mask ready. If you're going to go to a supermarket and you see that you have to be waiting in line, I would put on a mask. And then if you're immunocompromised or you're elderly, it's probably a good idea to put on a mask anytime you're in public, just in case the guy who's jogging by you breathes on you. So, so once the infection rates climb, then it's useful for everybody in public. And then they're absolutely recommended if you're sick, because they block the exhalation of viruses from a sick person. So again, the only controversy of mask is due to its limited supply. But I think people are wising up to this. The former FDA Commissioner, Scott Gottlieb, just tweeted his support for mask use. So I think you can only deny the facts for so long. So this is sort of an example of public health officials giving people partial information that's deliberately misleading. So I think they've been disingenuous, and it's caused people to lose trust in them. You know, they should just be honest. You know, people are adults. If you tell them masks are effective, but the rate of infection right now is so low and we don't have enough masks to go around that we'd like you not to use them, not to buy them. That makes complete sense. But telling people you shouldn't use them because they're not effective for you, it's true, but it's true only in a very misleading sense. And it doesn't engender trust when people find out the full truth. So you just need some filtration. So if you don't have a mask and the guy next to you is coughing, then just put your just put your nose into your sleeve and breathe through that. I mean, I do that when I go to the bathroom anyway, if it's a really smelly bathroom. And you know from experience that that blocks odors, so it must be filtering something. And in fact, a study shows that a t-shirt blocks 70% of virus-sized particles, so that makes sense. So I won't go into this, how to use masks and how to conserve masks, because I think most people just want to know whether it's a good idea or not. But I would say that in Asia, like in Taiwan and South Korea, China, and Japan, it's not just okay, it's expected that you wear masks. And in mainland China, it's required that you wear masks in public. So even if they have a very low infection rate, they have enough masks to go around. Yes?
Michael, for what's worth, we're trying to mobilize our alumni network to help us with masks, because I think that's, you're right. It's important.
Yeah. I mean, the US, the US is increasing its manufacturing of masks, but it's still slow at the moment relative to need.
So the washable masks are not as good as the N95, but they're still good.
Well, the surgical masks and N95 masks are equally unwashable. There's cloth masks that you can buy, like allergy masks. And again, they're probably better than nothing, but they're not as good as surgical masks, which are the 3-ply, flexible ones, and then, or the cup-like N95 masks. But on this slide, I say that I suggest decontaminating it by heating it up in the oven for 70 degrees Celsius for 30 minutes, or putting them in a UV sterilizer. But as we mentioned before, if you just let them sit in a clean place for a week, the virus should be dead. So I have a list of things that I think are okay to do, like visiting relatives if you're really careful with the hygiene steps. But I would definitely recommend buying groceries online instead of in a grocery store. You could say, well, the person who picks and packs your grocery might be infected, but that same is true in a grocery store. The person who stocks the items on the shelf has the same chance of being infected. Plus, in the grocery store, you have 1,000 other shoppers going through, and then the cashier has to touch the goods that 1,000 other shoppers have selected and touched. So it's better just to reduce the number of those interactions and just buy your groceries online if you can. But that's about it. So, travel if necessary. I think if you're armed with this, so if you're armed with hand sanitizer and mask, you can travel. Just be careful with, with keeping your hands clean, keeping your space clean. And I would just end with this, that because 50% of people with virus have no symptoms, but will become immune, and because that means 95% of people don't go to the hospital, most people who get the disease will recover at home in about a week. The workforce is not threatened. So, there'll be enough farmers and truck drivers and store workers to distribute food and sell food, so you don't have to go out and buy everything in sight. So that's about it. And in the next slide, as you guys are seeing online, there's a recipe for how to make your own hand sanitizer if you're not able to buy any, out of just ethanol or isopropanol and glycerol. So that's it. So I'm sorry to take so long. And
Michael, this has been fantastic. Thank you so much for taking time for us. I wonder, do you have a couple minutes for a few more questions, or?
Sure.
Okay, so we don't want to impose on your time, but we do have a couple of questions. Actually, we have a lot of questions. Let me just pick on a few.
Sure.
Can you talk about the possibility of reinfection?
Yeah, you mean like getting it infected again after you've cleared it?
Yes.
I think it's small. There was this worry that was because a woman had recovered from the virus and then left the hospital with a negative test, and then got worse and then got a positive test. and it was taken as evidence that you could clear the virus and get reinfected. But it's probably just the negative test was negative. I mean, she probably was fighting the virus pretty well, but then the virus rebounded. So it's more like a case of just insufficient immune response to the virus to begin with. But if she cleared it the second time and is really clear, then it should be fine. Your memory, your B and T cells take a week to ramp up, and but then your long-lived memory B cells take something like a month to differentiate. So there's this, it has to do with the switching of the types of Ig of immunoglobulin. So there might be, there might be this window where the virus can come back, but it's not because it's less likely that you're getting reinfected from outside as it's just a low-level infection inside.
So along similar lines, what can you share about the possibility of recovering this summer and fall? All the students return to school, and then we have a recurrence that's even worse than what we have now.
Yeah, so I think one thing that public officials are not saying yet, well, they don't know what's going to happen, but is that these kinds of measures are going to have to be kept in place until a vaccine or a very, very effective treatment is made. And so, the RNA vaccines, we'll probably know if they're useful in 6 months, but then it might take another few months to ramp up production and distribution. So we're not likely to be in time for a September vaccination. These drugs like chloroquine, remdesivir, chloroquine you don't want to really dose to everyone. It's too toxic for that. Remdesivir, you might dose it to high-risk populations. Remdesivir is likely to be in short supply. It's probably only 50% effective anyway. And so you don't want to rely on the drugs too much entirely.
What are
So
the
we'll have to have some
barriers?
restrictions.
What are the barriers to manufacturing these drugs faster and in larger quantities?
Chloroquine, there's no barrier. It's very simple. The favipiravir also looks pretty simple. I'm pretty sure they could be manufactured for millions of people, but it's probably the side effects are probably such that you don't want to take it if you unless you really have to. And the remdesivir looks more complicated. That's probably harder to synthesize, but Gilead is upping the production. But again, these drugs are not going to cure everyone of disease. And so they're more like hospitalization medicines. So they're only for the 5% of people who go to the hospital. And then maybe only 50% of those who take the drug will still be able to fight it off. And that's just a guess at the moment, maybe 75%. So it's still going to be a burden on the hospitals unless we continue to be careful. What I would like to see is us returning to work, but just being more careful like people are in Asia. I think it's a public education problem. In the US, the local authorities don't even bother trying a course of education before a course of lockdown because they've seen how people don't react to proclamations of keeping staying away from crowded places. So but in Asia you can do it. People are maybe much more scared of viruses, more used to working in tight environments, used to things like wearing a face mask and sanitizing spaces.
Can you also address briefly the testing bottleneck and what we can do to test more? I admire how Korea tested so many people, and they have tests that come around in 15 minutes, I hear. What
Right.
can we do here?
Yeah, I don't know. Yeah, I'm not sure about the 15-minute test. I really don't know. Maybe there is, but that would be amazing. And what I heard, what I saw in articles about Korea is that they, first of all, the companies took their own initiative to develop tests early, and then the health ministry coordinated with many companies to roll out multiple tests at once because they knew it might be a big thing and they wanted to be ready. Korea gets a lot of visitors from China, so they probably suspected they would get a lot of infections from China. In the end, they got most of their infections from one patient, patient 31, who infected people through her church. But they were ready for that. In the U.S., part of it is the federal nature of the government. Korea is still a relatively small country of 40 million people. So what's funny is, when China locked down Wuhan in the province of Hubei, that was 50 million people. And I just read an interview by Dr. Fauci where he was asked, would that ever happen in the U.S.? He's like, no, that's impossible. We could never do that. So now we've locked down California. That's 40 million people. It's the size of Korea. So it's possible to do it, but in the U.S. we have 320 million in the federal government. So and as we know, we have a federal government that's been, one, starved of resources, and two, ideologically reluctant to spend a dime on any public good. So it's just a complete failure of our political system. Well, I mean, let's be honest, it's a complete failure of one set of people in charge.
Michael, you have a lot of captive computer scientists and engineers on this call. What can computing and engineering do to help with this difficult moment in time?
Yeah, that's a good question. Not being a computer scientist myself, I don't want to give advice. I think you guys will figure it out. It's making cheap ventilators is certainly one possible solution. Using computer science to work with biologists to screen for drugs or analyze genomes is another possible solution. I think talk to your colleagues. Talk to your colleagues in biology. It's harder to do that now that we can't gather in the cafeteria. But we'll have a few weeks of being at home. It's possible to urge your department chairs to send messages to their other colleagues in other departments, asking if anyone needs any assistance in their research efforts.
Michael, thank you so much. If you get any computational challenges, please reach out to us. We would love to help, and really appreciate you sharing your time and your knowledge with us. Thank you.
Yes.
A clap on behalf of everyone.
Well, thank you very much. Yeah, appreciate your time.
Thank you so much.
Okay, signing off. Thank you.
Bye, everyone. Thank you for joining us. I hope you enjoyed the presentation and you learned